you that amino acid on the basis of their synthesis can be categorized into various families, and there are almost 6 families. Okay, first is the glutamate family, second is histidine biosynthesis occurs separately, hence it put into the separate class. The third is serine family, fourth is aromatic family, fifth is pyruvate family, and sixth is aspartate family. Okay, and we will see the biosynthesis of all of these family members along with regulation of amino acid biosynthesis and disease which can happen due to defective pathway, and then we will sum up this topic at the end. In basically, you all know about this, that the intermediates derived from the amino acid metabolism get entered into the glycolysis and citric acid cycle and pentose phosphate pathway for further metabolism. So all we already discussed a lot about this, so I'm not going to in much detail for this. And these are amino acids. This is the overview slide. Okay, like glucose, we all know about that. Glucose is converted into glucose 6 phosphate, and then glucose 6 phosphate is entered into pentose phosphate pathway to produce ribose 5 phosphate. Okay, this ribose 5 phosphate is involved in the synthesis of histidine, and this glucose 6 phosphate can lead to generation of 3 phosphoglycerate, as we saw just before this class, that this 3 phosphoglycerate can generate serine. Serine can be converted into glycine, and glycine involves in the synthesis of cysteine. So this is serine family. This 3 phosphoglycerate can convert it into phosphoenol pyruvate, then pyruvate, and this pyruvate involved in the synthesis of alanine, valine, leucine, and isoleucine. Here, erythrose 4-phosphate, which is generated through pentose phosphate pathway, can become a precursor for the synthesis of phenylalanine tryptophan, and phenylalanine can produce tyrosine. This citrate, it can act as a substrate for the synthesis of oxaloacetate and alpha ketoglutarate. Okay, and this oxaloacetate and alpha ketoglutarate involves in the synthesis of asparagine, arginine, lysine, threonine, methionine, glutamic acid, glutamine, proline, and arginine. Okay, so this is all about the overview. What are the precursors and how they involve in the synthesis of a particular amino acid, we will see in the next slides. This is the first family of amino acid biosynthesis, that is the glutamate family. Okay, here the key ingredient is alpha ketoglutarate, which on transamination reaction in presence of enzyme aminotransferases can transfer its amino group to the amino acid. Okay, and if 2 molecules of alpha ketoglutarate interacts with each other in presence of aminotransferases, it produces one alpha ketoglutarate and one glutamate. And this glutamate, basically in presence of enzyme glutamine synthetase, which requires 1 ATP for reaction, get converted into gamma glutamyl phosphate intermediate. Okay, and this gamma glutamyl phosphate intermediate interacts with ammonium ion and produces glutamine. So basically, this is the overall reaction, how alpha ketoglutarate converted into glutamine. Okay, so glutamine is the basic transporter of ammonium ion into the cell. It supplies the ammonium group in the variety of reaction, because ammonia is toxic to cells, so it cannot move freely inside the body. It get incorporated into glutamine and transported to liver for further detoxification reaction. So how this glutamate or glutamine involved in the synthesis of other amino acid like arginine and proline. This glutamine glutamate converted into N-acetyl glutamate when it reacts with acetyl coenzyme. And this N-acetyl glutamate converted into N-acetyl glutamate 5-phosphate, which upon dehydrogenation reaction converted into N-acetyl glutamate 5-semialdehyde, and this N-acetyl glutamate 5-semialdehyde converted into acetyl ornithine, then to ornithine. And this ornithine basically interacts with glutamate 5-semialdehyde and another molecule and produces pyrroline 5-carboxylase that ultimately forms proline. So this is the basic pathway for proline synthesis, and this glutamate can glutamate can convert it into glutamate 5-phosphate, then into glutamate 5 semialdehyde, and this semialdehyde get converted into pyrroline 5 carboxylase, which again converted into proline. How histidine synthesized from ribose 5-phosphate. Okay, ribose actually 5-phosphoribosyl alpha pyrophosphate provides 5 carbon atom for purine biosynthesis. So it is very important reaction, and this 5-phosphoribosyl alpha pyrophosphate actually involved in the pyrimidine biosynthesis also, and it is also involved in purine. It is also involved in purine salvage pathway, and it is also acts as an intermediate in tryptophan biosynthesis. Okay, so glutamine also supplies the second ring nitrogen to the purine. Okay, and this alpha D ribose 5-phosphate from this shunt, histidine shunt, it is basic precursor for all other metabolic reactions. So this ribose 5-phosphate basically involved in the synthesis of histidine. This is the overall reaction. I know this is a complicated one. So I'm just tell you the key reactions of this pathway to just memorize you how it get synthesized, how histidine gets synthesized. Just see this. You all are well aware of this, as I think Dr. Munjal sir had taken one lecture on purine pyrimidine biosynthesis earlier. Okay, so you just correlate this from there. 5-phosphoribosyl pyrophosphate, this is PRPP. PRPP, actually this is the structure of 5-phosphoribosyl pyrophosphate, where you can see this is the ribose unit. Here one phosphate group is attached at this carbon and 2 phosphate group is attached at this carbon. Okay, and this interacts with ATP and forms N5-phosphoribosyl ATP, adenosine triphosphate. Okay, it get attached to this group, this carbon, and which upon subsequent removal of pyrophosphate group forms 5-phosphoribosyl AMP. And this 5-phosphoribosyl AMP, we will also see this pathway in our nucleotide biosynthetic unit, and the same pathway we will learn there from scratch. So don't worry about it, how complicated is this. And this is very simple. Okay, this phosphoribosyl AMP interacts with another molecule and get converted into an open chain compound 5-phosphoribosyl 4-amino 5-amino imidazole D carboxamide ribonucleotide, which reacts with glutamine, and here glutamine supplies amino group, and here 5-amino imidazole 5-carboxamide nucleotide get removed, and leftover is their imidazole glycerol phosphate. And this imidazole glycerol phosphate, upon dehydration, one water molecule removes, get produced imidazole acetol phosphate, which again reacts with glutamate and one amino group transfer here, and it forms histidinol phosphate, and which upon removal of phosphate group produces histidinol. Here it is known as histidinol because OH group is attached. So again dehydrogenation reaction occurs here, and this OH group is converted into carboxylic group, and this histidinol becomes histidine. So this is overall biosynthetic pathway for histidine. It basic precursor for this biosynthesis is PRPP, that is 5-phosphoribosyl pyrophosphate, which again subsequent interacts with ATP and involves in 4-5 reactions and leads to the production of histidine. You can see the steps clearly written at the bottom of this slide. First step is ATP phosphoribosyl transferase. The second enzyme is phosphorylase. The third is phosphoribosyl AMP cyclohydratase. This is the cyclohydratase step. And the fifth is glutamine amidotransferase. Sixth step is imidazole glycerol 3-phosphate dehydratase. Seventh is L-histidinol phosphate aminotransferase. The eighth step is phosphatase, as I said, and the ninth step is dehydrogenase. Then our histidine get produced. So remember this pathway. The next is serine family. For the biosynthesis of serine, the basic precursor is 3-phosphoglycerate. We already have seen this. Here an enzyme 3-phosphoglycerate dehydrogenase or dehydrogenase convert this 3-PG into phosphohydroxypyruvate, and this phosphohydroxypyruvate interacts with glutamate and produces phosphoserine, which upon undergo a reaction catalyzed by phosphatase and produces serine. We have seen it in our earlier class, and this serine can either convert it into glycine and or it can involve in the synthesis of cysteine. We have seen this reaction. I'm just quickly going through all the reaction. 3-phosphoglycerate interacts with NAD and forms, in presence of dehydrogenase, form 3-phosphohydroxypyruvate, which interacts with glutamate. Amino transferase reaction happens here, and it forms 3-phosphoserine, which undergo phosphatase reaction and produces serine. This serine can transfer its methyl group to tetrahydroxyfolate pathway by a transferase reaction and produces glycine. And how cysteine biosynthesize in the body? It synthesizes from homocysteine. Homocysteine, when reacts with serine, it forms, in presence of synthase enzyme, it forms cystathione, and cystathione lyase catalyze the removal of ammonia from here, and it produces alpha ketobutyrate and cysteine. So this is the pathway for cysteine biosynthesis. Another family is the aromatic family. Here the biosynthesis of aromatic amino acid like phenylalanine, tyrosine and tryptophan occurs in plants and microorganisms, but in animals these are categorized under essential amino acid. We have to take it into the diet. Okay, how they get synthesized? Basically phosphophenol pyruvate, when it interacts with erythrose 4-phosphate, which is generated from HMP pathway, when they interact, they form phenylalanine, tyrosine and tryptophan. Phenylalanine can back converted into tyrosine. Basic intermediate in this pathway is corishmate, and corishmate is important. Remember this term. And this also known as the shikimic acid pathway. If you learn deeply the plant biosynthetic pathway, phenol biosynthetic pathway in plant, you should be aware about the shikimate pathway or korishmate pathway. Okay, so these are the basic biosynthetic pathway in the body. Basically, phosphoenol pyruvate reacts with erythrose 4-phosphate and produces, in presence of a synthase enzyme, and produces 2-keto 3-deoxy D-arabino haptulo sonate 7-phosphate. Okay, and this compound basically reacts with another synthase enzyme where phosphate group removes and produces 3-hydroquinate, and this 3-hydroquinate forms 3-hydroshikimate, and this 3-hydroshikimate basically converted into shikimate. This is the structure of shikimate, which in presence of kinase enzyme produces shikimate 3-phosphate, and which again interacts with phosphoenol pyruvate in presence of uh, synthase enzyme and forms 5-enoyl pyruvyl shikimate 3-phosphate, which upon removal of one inorganic phosphate in presence of enzyme korishmate synthase produces korishmate. Okay, so this is the overall pathway by which korishmate gets synthesized, which is a basic precursor for further other amino acids. Like korishmate interacts with glutamine and produces anthranilate. Anthranilate, okay, and this is the anthranil. You have seen this compound in laboratory, okay. This anthranilate interacts with phosphoribosyl pyrophosphate (PRPP) and produces 5-phosphoribosyl anthranilate. Okay, this 5-phosphoribosyl anthranilate converted in a enol 1-alpha carboxyphenyl amino deoxyribulose phosphate, which actually undergo a decarboxylation and dehydration reaction and produces indole 3-glycerol phosphate. This indole 3-glycerol phosphate actually from this glyceraldehyde 3-phosphate get removed, and it produces, it interacts with a molecule of serine and it produces tryptophan. Okay, so these are all reactions how korishmate get converted into tryptophan. You have to just quickly see these reaction and just remember the basic intermediates of each pathway. Like korishmate is the basic intermediate for tryptophan biosynthetic pathway. How tyrosine and phenylalanine formed from korishmate. Korishmate can be converted into prephenate. Actually, the enzyme involved here is mutase, and this prephenate in presence of a dehydrogenase enzyme get converted into 4-hydroxyphenyl pyruvate, and this 4-hydroxyphenyl pyruvate interacts with glutamate in presence of aminotransferase and produces tyrosine. And in the parallel reaction, this prephenate in presence of dehydratase enzyme, which removes one CO2 and hydroxyl group, produces phenyl pyruvate. This phenyl pyruvate interacts with glutamate and produces phenylalanine. So this is the overall biosynthesis of tyrosine and phenylalanine from korishmate. Coming to the next family, the aspartate family. Aspartate family actually belongs to the family where synthesis of aspartate and their colleague amino acid happens in this pathway. Basically, aspartate synthesized from oxaloacetate through a simple aminotransferase reaction. Here, glutamate can be a amino group donor. It transfers its amino group to oxaloacetate and get converted into aspartate. This aspartate again interacts with the glutamine and takes its extra amino group and get converted into asparagine. So in this way, aspartate and asparagine synthesis is very simple and easy, and you can understand it easily. Aspartate is non-essential amino acid, and one-step transamination reaction can lead to its synthesis. Okay. But remember this thing, glutamate is always a amino group donor for aspartate and asparagine synthesis. How methionine, threonine and lysine get synthesized? Aspartate is common precursor of these essential amino acids. The committed state is conversion of aspartate to aspartyl beta phosphate, and the branch point, branch point occurs at aspartate beta semialdehyde step, and we can see this through a reaction pathway. You can see this here. Aspartate can be converted into aspartyl beta phosphate in presence of ATP, and this aspartyl beta phosphate is converted into aspartate semialdehyde, which upon addition of pyruvate forms 4-hydroxy tetrahydrodipicolinate, and this 4-hydroxy tetrahydrodipicolinate converted into dihydrodipicolinate, and this will back converted into tetrahydrodipicolinate, and this tetrahydrodipicolinate interacts with succinyl CoA and produces succinyl 2 amino 6 keto pimelate. Okay, this pimelate interacts with glutamate and get converted into N-succinyl diamino pimelate. This N-succinyl diamino pimelate converted into diamino pimelate through removal of succinate and addition of water. This compound back converted into meso alpha epsilon diaminopimelate, which is which upon removal of CO2, this is decarboxylase reaction, get converted into lysine. In the subsequent parallel pathway, the beta aspartate semialdehyde can convert into homoserine. This is dehydrogenation reaction, and homoserine can convert it into phosphohomoserine, and this phosphohomoserine can convert it into threonine. And here homoserine can also convert it into orthosuccinyl homoserine, which interacts with the cysteine and produces cystathione, and this cystathione can, in presence of lyase enzyme, can be converted into homocysteine, which interacts with the tetrahydrofolate intermediate pathway and produces methionine. So this is the pathway, basic pathway, how threonine, methionine and lysine generated through aspartate. The next family is the pyruvate family, where the alanine pyruvate can be can involve in the synthesis of alanine, valine, leucine, and isoleucine. Alanine is non-essential amino acid. Simple transamination reaction can convert pyruvate into alanine. Just removal of this keto group and addition of an ammonia here can convert pyruvate to alanine. And here glutamate can donate this amino group, and it after donation it can convert it into alpha keto acid. How leucine, isoleucine and valine forms from this? Actually, pyruvate, when interacts with TPP, it forms hydroxyethyl TPP, and this reaction, we can see this via this schematic more clearly. Actually here pyruvate interacts with TPP, thymine pyrophosphate, and forms acetyl thymine pyrophosphate unit, which interacts with alpha ketobutyrate and forms alpha acetohydroxybutyrate. This alpha acetohydroxybutyrate get converted by a subsequent reaction, get converted into isoleucine. Okay, and similarly this compound can also interact with another pyruvate and forms alpha acetolactate, which through subsequent reaction can convert it into valine. And this intermediate of this pathway, valine biosynthesis pathway, alpha keto isovalerate, can involved in the synthesis of leucine pathway. So this is the overall biosynthesis. You can see here at every step There is just dehydrogenation, phosphorylation, or addition of small molecules or removal of carbon dioxide. And the overall, the end product is our desired amino acid. Here, if you want to learn it in detail,